First up is Dr. Bingqing Liu from she's an assistant professor at the University of Louisiana at Lafayette. She specializes in satellite oceanography, and she focuses on coastal carbon dynamics, aquatic biodiversity, and water quality, and their responses to climatic and anthropogenic stressors. So she's going to talk to us today about the synergy of artificial intelligence and hyperspectral remote sensing for coastal water quality monitoring. And do I need to switch the slides? I'll switch the slides real quick. I think that's good to switch to display. Awesome. Well, thank you. Well, welcome.
Thank you so much for the introduction, and thank you for having me. This is a really great opportunity, and it's so wonderful this morning and catching up so many fantastic updates on the PACE application and research. Today, my talk is going to focus on the synergy of AI and PACE for coastal water quality monitoring. So to begin, I'd like to share this background photo that taken along our coast in Lake Pontchartrain, one of the biggest estuarine lakes in Gulf Coast, and it highlights a toxic cyanobacteria bloom in large scale. And I thought it is a common issue that we are facing in our estuary and coastal systems because of the eutrophication, and it always causes high standing stock of phytoplankton, sometimes leading to harmful algal blooms, and further the hypoxia events, like in the northern Gulf of Mexico, hypoxia event occurs every year in either large scale and small scale, and also expanding into the estuaries, threatening the fish and the shellfish habitats and industries. And this water quality issue is going to become worse because of the warming climate and continuous anthropogenic factors. So there's interdisciplinary interest in enhancing the monitoring and the forecasting capabilities about coastal water quality, and the PACE is playing a very important role in that. Actually, this is a slide I borrowed from Morgan about the RSAT training using PACE data, and it's short and sweet, summarized our work as early adopter, PACE's early adopter, and the science team members. And we're trying to use PACE data to look at coastal water quality, specifically phytoplankton community composition for current and future scenarios using PACE, AI, and the numerical model simulations. And we work with our end users. I'm trying to get them to know PACE, understand PACE, and use PACE data easily. So we had done the first successful step, and we developed and published a Python package tool called HyperCoast, and it works for PACE and as well as other hyperspectral missions. You can easily download and open up, extract the spectrum from hyperspectral remote sensing data, and welcome to explore. So before I dig into our work, I just want to give a little bit background about ocean color remote sensing. I guess most of you are already familiar with this, but those are fundamentals, and we can see a RGB PACE composite imagery after Hurricane Milton in the Gulf of Mexico. It's so colorful because of these optically active constituents in the water, like sediments, color dissolved organic matters, and phytoplankton. A large-scale bloom happened along Florida coast. And it is colorful because of absorption and backscattering properties of these constituents, and they together determine how much light leaving the water surface and further determined by the satellite. Not somehow, and they can link the two remote sensing reflectance through the simplified radiative transfer model. And this is a very simplified version, but it tells us that whenever we have remote sensing reflectance, we have chances to estimate the absorption and backscattering properties. It's part of our work and very important related to the biogeochemical parameters. And this is the radiative transfer model. And if you know IOPs, and it will be easier for you to calculate the AOPs, apparent optical properties, like remote sensing reflectance. But if you want to estimate IOPs from remote sensing reflectance, it's inversion problems more challenging in coastal waters because sometimes the sediment and color dissolved organic matters in water overwhelm the phytoplankton optical properties. And also the combination of these IOPs along with their concentration sometimes can give you similar or same remote sensing reflectance spectrum. And also, if you compare the multispectral and hyperspectral, you know that multispectral in the past missed a lot of details about the aquatic environment. So here I want to show a demo of paste data. It's interactively displayed using HyperCoast tool. And again, this is an image acquired along Florida coast after Hurricane Milton. And this is a mixed bloom for the red tide species, Crania brevis, and Coccolithophore. You can see clearly the phytoplankton signature there, and also the sedum-rich waters. And if you compare the red tide area and the Coccolithophore, they showed so much difference for their spectralship. And this is the powerful of PACE. It tells you so much details about phytoplankton community structure. Here, the left panel, it is also the spectral way extracted from PACE, and clearly you can see cyanobacteria signature. And our question is how we can develop algorithm that translate this diverse spectrum into plain language, like what type of algae they are, how abundant they are, potentially whether they're toxic or not, and we want this algorithm not being limited too much by specific time in the region. But phytoplankton, they're so diverse. They have different size and morphology, and they have different biogeochemical functions, and they respond to climate change differently. And one of the optical proxy we used is phytoplankton absorption. And as you have seen here, they absorb light differently because they have pigments in their, different type of pigments in their body. And that's why they show different colors when you observe cultures in lab or they're in bloom conditions in the field. So to predict the absorption properties from remote sensing reflectance is not an easy task, and I really appreciate previous efforts synthesize these water quality data sets and hyperspectral remote sensing reflectance along with absorption properties. And in our lab, we also do some lab experiments to characterize some species and genera and groups that we are interested in for their absorption, backscattering, and reflectance. Like here, it is an example that we look into. This sample is very turbid if you look at it, but under the hyperspectral microscope, it allows us to look at the phytoplankton cell down to nanometers, and you can clearly see where sediment and where cyanobacteria cells, and even within cyanobacteria groups, you can see the difference of the spectrum for the reflectance. And we plot them on PACE wavelengths. That means PACE also have the capability to differentiate different type of phytoplankton. So with better characterized IOPs and based on the radiative transfer model, and we can simulate hyperspectral data along with the field data, and so we're really trying to use this data set to feed AI models. To me, AI and open science, they seem twins, and because the foundation of the AI is data. After we generate this synthesized data set, and we developed different AI-based models. And here's an example. It's called a variational autoencoder. I won't dig into details, but it's very practical and good for the hyperspectral applications. And we're trying to predicting phytoplankton-related parameters like chlorophyll A and absorption from remote sensing reflectance. And here is a performance of this model in predicting phytoplankton absorption. And we have done tons of field work in the Northern Gulf of Mexico to validate the performance of the model. But we welcome the collaboration, so we really want to validate the model outside of the Gulf of Mexico. And we share all the code in HyperCoast, and welcome to explore, and it's available for us to hear your feedbacks as well. So this is an example of chlorophyll A products in the Northern Gulf of Mexico, and we have done some field work to validate its performance in different estuaries, like in the lake, in estuaries, and in coastal waters. And so far, its performance is accessible, and we just need more data to understand the uncertainties in coastal waters. And why we're doing that, because sometimes the algorithm for open ocean typically does not work well in coastal waters and inland waters. In addition to chlorophyll A concentration is the basic parameter describing the abundance of phytoplankton. We mentioned that we're also curious about the phytoplankton absorption because it has so much information about the structure information of the phytoplankton community. And the top panel of the figure is a comparison between field collected RS remote sensing reflectance and the PACE reflectance. And you can see PACE is so powerful to capture, wonderfully capture the spectral shape of remote sensing reflectance, but for the magnitude, it seems over the estuarine waters, especially in the sedum-rich waters, and its blue wavelength just seems corrected too much, and there's offset there. And based on that, and also that model, we predict the phytoplankton absorption spectrum, and this is a comparison between field and the predicted spectrum. And again, the spectral shape is good, and the magnitude is the issue right now, and we're trying to tune in this model. And why we want absorption so much for phytoplankton, and because we can go further. And this is one of the approach that you can look into phytoplankton community structure. And we decompose the predicted phytoplankton absorption into different phytoplankton group. Of course, you need to have some pre-knowledge, like what type of phytoplankton group is dominated over there. And here we selected 5 diatom, chlorophytes, cryptophytes, dinoflagellate cyanobacteria. And you can see actually the weight of each colored line here is chlorophyll A concentration of specific phytoplankton group. Obviously here the diatom is dominated one with the highest chlorophyll concentration, and the diatom and chlorophyll, these seems share the similar spatial distribution, and for cyanobacteria and dinoflagellate, they share the similar spatial distribution, and it ecologically makes sense to me. And the diatom chlorophytes, they seems in more nutrient-rich waters and more turbulent and close to the distributaries, and then cyanobacteria and dinoflagellate is in the middle of the lake. And we have not quantitatively analyzed uncertainties of the algorithm. That based on the preliminary results from HPLC, you can see it ecologically makes sense. Like here, the high fractions of cyanobacteria in the middle of the lake, and if moved to 8 and 9 the sites, and you can see it's close to the freshwater input, where we saw the high fractions of diatom and the chlorophyte. So this type of products is not only used for like harmful algal bloom studies, and we care about it because phytoplankton is the basis of food web. And it helps us to understand better the diversity, biodiversity, and aquatic food web. So we work with our end users, including Coastal Protection Restoration Authority, Louisiana Department of Environmental Quality, and the EPA National Estuary Program, and the Department of Wildlife and Fisheries, as well as Pontoon Conservancy. I just want to show an example how we work with the end users. And in Louisiana, our wetland loss at alarming rate. And if you compare, I just back and forth, you see the land loss and predicted use our model in future 50 years. That tells us in future we're gonna have more estuaries, the shallow water area, and because of that, our state put a lot of efforts on the restoration. There's a giant restoration projects on the ground or to be underground. And one is very interesting, it's called sediment diversion. And we divert fresh water into estuaries, and it's sediment rich. It can nourish the wetland, reduce the wetland loss rate. But in the meantime, nutrient into the estuaries, it causes water quality issues. And using PACE or some other hyperspectral missions, we can monitor the current conditions of phytoplankton community composition. But it's hard to tell us what if the restoration project on the ground, what's going to happen. So we try to integrate PACE and other satellite data as well as numerical model to look at the future scenarios in the face of climate change as well as the restoration project. And this is current the model we have, and we leverage NOAA hypoxia project to set up this model. And for most of the biogeochemical model, they only have like large, small phytoplankton. We are trying to expand the model by including more phytoplankton groups or species using PACE data, and to inform the model and validate the model, and to achieve the 3-dimensional prediction of phytoplankton community structure for future. Well, this is pretty much I want to cover for my talk, and I'm ready to pick your questions.
algorithms with remote sensing transfer. You had some linkages in terms of in your model, and you called them something specific that now I'm forgetting what they're called. You called them encoders, right? And so linking those encoders between remote sensing reflections, so AOPs and IOPs, and then AOPs, IOPs with the biogeochemical constituents. In time, those are, and especially in coastal areas, it's very non-stationary. So I'm wondering how we as a community kind of merge the best of AI and our kind of traditional models, radiative transfer, et cetera, to maintain mechanistic connections between those AOPs, IOPs, and BGCs?
Thank you so much for your question. And I think my team member may be in a better position to answer this question, but I will try to answer. So for our work, we know that in estuary and coastal zone, we need a specific algorithm based on like a red or near infrared absorption or reflectance properties, and to generate some empirical relationship instead of based on the blue-green region for open ocean. But also across the regions and across the times, and this is not a simple relationship. And also considering there are so many stuff in the waters, and sometimes if you have same chlorophyll A concentrations, you might have different remote sensing reflectance spectrum. So this is why you need to use AI, and they can handle complex relationships. And in the meantime, we move into hyperspectral. You need to consider the computation resources. You need to select an algorithm that's practical for hyperspectral remote sensing. And that this is the one part we have been exploring. What could be more stable and more practical for hyperspectral predictions. And for the uncertainty analysis, and we train the model using the simulated data sets. We hope, because it's a more comprehensive data set, remember that we have absorption spectrum here, and it's from other study, and we're trying to use different combinations of these 17 phytoplankton species, and to generate a comprehensive data set to cover different type of spectrum, which we don't have in the field. And so we let the model to learn the simulated data first, and it learns something, and it transfer what it learned from simulated data set, and then we use field data to fine-tune this model. And somehow, I think for practical application of AI algorithm, it really requires the fine-tune process. You need to have some like local knowledge to fine-tune or downstream your add some downstream tasks for your AI models, and to reduce the uncertainty. And in the meantime, avoid over-tuning of model. Well, I think I'm not a very good position to answer this question, but I tried my best. But you can always reach out to us, and our paper is under review, and hopefully more details will coming out. All right, thank you.